Welcome, everybody. Thank you for making it out to seminar this afternoon. I am pleased to invite or introduce our speaker for today. This is Dr. Ben Parrott. Ben is an associate professor at UGA Odo School of Ecology, and he's also faculty at Savannah River Ecology Lab, so he's based in Aiken, South Carolina. Ben has a bachelor's in biology, a PhD in cellular biology, and he did a postdoc at the Marine Biomedical and Environmental Sciences program at MUSC. And there he actually studied under someone named Lou Gillette, who you may not know, but he is broadly considered the father of crocodilian and alligator endocrinology, had a big impact on the field. And I had a great pleasure of meeting Ben through my work with American alligators over the last several years, and I think he does some really exciting work that you'll hear about today. So without further ado, I'll go ahead and turn it over to him and let him share his research with you.
Thank you. All right. All right. Thanks for the let me make sure I got this going. Okay. Thanks for the invitation. Thanks everybody for showing up. Huge honor to get to come here today and share some of the research that we've been working on, over the last several years. And it's been fun. So I got I was on the way here, and it's only about a 2.5-hour drive from the Savannah River Ecology Lab. I was thinking, I wonder why I've not been here. I've been here once when I was a postdoc for a developmental biology meeting. They kind of had us like squirreled away off campus somewhere. It was on the lake. It was nice, but didn't get to see campus at all. So Kathy showed me around, and neat to see I feel like I'm kind of behind enemy territory here, behind enemy lines in a way, but feels good. So what I thought I would do is I'm going to share a project that's kind of two projects. It's centered around what I call ecological developmental biology. And I'm going to place this within conservation frameworks, and then a little bit of the work we're trying to do in terms of placing this in an evolutionary framework. But before I get into it, I wanted to acknowledge some co-authors here. So Samantha Bach is a graduate student in the lab, and she's going to be graduating or defending later this month, actually. And Thomas Rainwater is part of this unit, and he's at the Peru and works at the Yaqui Wildlife Center. But I can tell you all the work that we've done, or that I'll show you today, definitely not possible without these two. And so I just wanted to get that out of the way really early. All right, so I wonder, can I make that thing go away, or do I need to?
If you click on the more dots and then hide floating eating controls down a few more, one more.
There it is. Okay, great. So I want to give a really kind of broad overview of the types of work that we do, because the work that I'm going to show you today is not really like the work we do. But we're generally interested in kind of pursuing this question of how complex environments become integrated into organismal phenotypes. And we spend a lot of time looking at reproductive biology, as well as more recently kind of the biology of aging. And even within kind of those spectrum of traits, there's a lot of variation and ways to think about that, whether you're thinking about reproductive fitness or life history and senescence, but also in biomedical context, right, where you're thinking about reproductive health and age-related disease. But in any case, I think over the last several years, I actually saw this week was the 70th anniversary of Watson and Crick's discovery of the double helix, right? And so not that long ago, really, but anyway, so over the last several decades, we've come a really long way in understanding kind of the genetic underpinnings and contributions of phenotypic diversity. And I think though, kind of what we're still coming to grips with, what we're still understanding in this post-genomic era, right? We've got genomes for all sorts of different species. We've got really cool tools we can use to study genomic processes. But I think what we're kind of relearning is that the environment is still going to be a large part of this, and that the genome is not going to be the whole story. Might not be the whole story. I know there's a delay or it's.
It's just not advancing.
It's not.
Try some of the arrow keys on the keyboard.
There we go. Okay. So whether it's communicable diseases or stress, exposure to contaminants or climatic variability, nutritional quality, social cues. I think what we're kind of trying to figure out is how are all of these factors kind of interfacing with themselves to make the environment, but then how are they also interfacing, particularly with developing organisms, and how are they kind of being translated into these biological pathways and genomic level processes. So we spend a lot of time thinking at this level here. But what I'm going to talk about today is our work kind of on the other side here, thinking about how some of these traits that are influenced by the environment scale and affect life history, as well as perhaps population dynamics and evolutionary dynamics. So that's kind of the general framework. We spend a lot of time doing molecular work, as I've alluded to, but I'm only, this I think is the only molecular thing I'll show today. And the only point I want to make here is that, so I'm going to talk a little bit about sex determination. And what becomes really clear is that there's this remarkable kind of conservation of these genetic pathways that build the testis or the ovary, the vertebrate testis and ovary. So it doesn't matter what these genes are, but they're highly conserved, and they're required to kind of build this architecture of the testis and the ovary, and at a histological level, you see that reflected, right? So like an alligator ovary looks very similar to a mouse ovary under a microscope, for example. But standing kind of in stark contrast to this conservation of core machinery is a really remarkable divergence in the way in which animals determine sex, right? Or the way in which we see sex determined. So we're probably most familiar with, let's see if I can put this in. With sex chromosomes, right? So the X and the Y chromosomes that we have, and Z and W in birds and other animals as well. But there's also polygenic systems, so you can have multiple loci that are located on different chromosomes, and they all kind of give this summary effect and get a threshold effect to get one of the two sexes. But there's also a lot of taxa that use environmental cues, and I'll get into that in a minute. But to make matters even more kind of complicated, there's cases in which you have environmental cues interacting with genetic systems. And so if you kind of look at this as a whole, what you see is that there's this kind of funnel where you see all of this conservation and how to build a testis and an ovary. That's, pretty similar, pretty conserved. But then you see all of this variation in the way that organisms determine sex. And so kind of extreme divergence there. And I'm going to come back to this in a minute, but what I wanted to do is focus on environmental sex determination. And in this particular slide has nothing to do really with the research other than it's environmental sex determination. And this is a really cool organism here. So this is a green spoonworm, marine species. And this picture doesn't do it justice, right? The anatomy, this thing's got this like body here and this long proboscis. And if you see a video of this thing, it's kind of like undulating and doing these really bizarre behaviors. But in this case, this is a female worm. And what she does is she releases marine larvae that are motile out into the ocean. And where those larvae end up will determine whether it develops as a male or a female. So, for example, if you have a larvae that gets released and it just ends up on the ocean floor, for example, it will develop as a female. And, you know, it will resemble the mother. And if if the larvae ends up coming into contact with another green spoonworm, it will actually migrate down this tube, down the proboscis into the body here and develop as a male. And it's this really reduced form. It lives in the body. It's females feeding it, and the only thing it needs to do, right, is just sit there and fertilize the eggs. So in this case, right, it's pretty bizarre, but the sex is determined purely by the social interaction, right? Does it interact with another green spoonworm or not? And this is quite kind of a bizarre and semi-unique occurrence, but the fact that environment is determining sex here is not bizarre at all. And we see all sorts of environmental factors that determine sex. So things like social status is pretty common in some fish. And they can actually undergo sex change, you know, not even during embryogenesis, but even after their one sex has been determined, they can reverse. But by far, temperature here is the most common environmental factor that's involved in environmental sex determination. And so I'm going to call this temperature dependent sex determination, or TSD. That's where this acronym right there. And it's really common in non-mammalian species, particularly vertebrates. And so this is gharial, and all crocodilians have TSD. So they completely lack sex chromosomes. There's no X, no Y. And temperature is the only known determinant of sex that we know of. Same with this kind of Yoshi-looking creature here, which is a tuatara that's found only in New Zealand, and it's the same way, only temperature. Most turtles, there are some genetic sex determination species, but most of them have TSD, including like all the sea turtles. But yet, you see this in some squamates. So this is a bearded dragon like you would find in Australia. They have a ZZZW system, but at certain temperatures, the sex can be reversed. So they have the ZZZW, but you can actually reverse their sex with particular incubation temperatures. And then in some species, like this is the European sea bass, it's polygenic, but you also have a temperature influence. And we think this is probably some sort of not a stable situation right there, but we won't get into that. So if you look at just the reptiles here, you see different kind of patterns of sex determination. And so in tuatara, they're kind of have their own thing going on here. But what I'm showing here, if you can not read this, sorry, is the sex ratio. So this is the percentage of males on the Y, and this is the incubation temperature here on the X. And so they have this female male pattern. So at low temperatures, you get all females, and then at high temperature, you get males. And all crocodilians have this pattern, which is a female male female pattern. So at low temperatures, you get females, and also at really high temperatures, you get females. But at these intermediate incubation temperatures is where you see the production of males. And then in turtles, most turtles, there's a couple like the snapping turtles, a little different. Most turtles, like this red-eared slider, has male-female patterns. So at low temperatures, you have males, and at high temperatures, you get females. So there's some variation there. I think it's kind of interesting to think about where these patterns might emerge and what drives them. But what I want to do is kind of use the alligator system today as a almost a case study or a model to kind of look at both how to approach TSD within this conservation framework, as well as trying to understand why in the world organisms would even use temperature to determine sex in the first place. So a little bit of kind of natural history about alligators. So this is a female with her nest here. So she has built this nest, and mating typically occurs in early spring, maybe late spring. So not now, but they're probably gearing up to mate. And, you know, you'll hear male bellows and stuff like that come April probably. And females typically lay no more than one clutch a year. So just one go, and Thomas Ringwater and some of his collaborators have shown that at least at this one site, you know, a female may only lay a nest or build and lay eggs once every 3 years on average. So, you know, it's a big investment. And what she does is she'll kind of take her legs, her back legs, her tail, kind of pile all of this vegetation up in this pile here. She'll crawl all over it, kind of pack it down, and it turns into the kind of glorified compost pile. And in June, so mid-June, you see the peak of oviposition. So she'll crawl up here, dig a nest cavity into the nest, oviposit about 30 to 50 eggs. And they'll stay there for about 63 to 75 days, depending on the temperature inside this nest. And then they'll hatch, and she'll in some cases help them get to water. So this is what it looks like when we go collect the eggs, right? So you kind of open up the nest, and you can just kind of get an idea of the size of the eggs there, and we can pilfer those eggs in some cases. So this is one of the things that the figures that kind of got lost in the transition from my computer to this computer. So there's a lot of other dots here that you can see some uncertainty here. What we did is we went and we found every paper that reported the incubation temperature and the resulting sex ratio, and we plotted that, and we kind of weighted it by sample size. But this is the trend that we see. And again, what you can see here is that these intermediate temperatures, so about 33 degrees, you'll get 100% males in most cases. And anything below 31, you get about 100% females. And why I think that's kind of interesting, that means that just a small difference in temperature, 2 degrees, can lead to these vastly different sex ratios, which can become important and will become important in just a little while. And this was the other thing I wanted to mention, was that the temperature acts within this very discrete developmental window to determine sex. So we used to think it was between Ferguson stage 21 and 24, which probably means nothing, the stages, but it's right when the gonad really starts to differentiate. So the gonad starts as this bipotential primordium, and then it starts to differentiate into either an ovary or a testis. And that's happening right at 21. But we were getting weird results all the time, where we would incubate and not get the sex ratios we thought we would get. And so we did these shift type experiments. So this was a male promoting temperature here, I think it was 33, and we got 96% males. But if we started doing these shifts even early, prior to this thermosensitive period, we would see this sex reversal, indicating that these embryos were sensitive to temperature much earlier than we thought. And so if kind of look at and do the logic there, what we really think is happening is this window is lasting from stage 15 to 24, which equates to about 23 to 28 days. So there's about 3 to 4 weeks where temperature is being experienced in kind of informing this sex determination system. And so one of the first questions that I wanted to share, or one of the first projects that I'll share, is centered on this question of what are the environmental and ecological influences on the nesting environment. And so all the work that I've shown you has been using constant incubation temperatures, right? And actually all the work that I'll show you after this is using constant incubation temperatures. But we don't really have a great sense of what the thermal environments look like in nature. And further, we thought we wanted to look at kind of what we're seeing in these natural nests and see if we could understand what the drivers of that thermal environment were, so that we might kind of start to ask, is there a potential for these skews in population sex ratios under contemporary climate change? So the general idea was that we would go out and find all of these alligator nests and stick thermal augers in them and get kind of a sense of what their thermal environments would look like, and see if we could model that, see if we could model, what are the drivers, and then start plugging in climate change models to ask what nests would look like in the future and how that might change sex ratios. So straightforward strategy here. And we did this at 2 different sites. So one is at the Yawkey Wildlife Center, which you're probably familiar with, right here on the coast of South Carolina. And the other was Kennedy Space Center, or Merritt Island, which is further south. And so we've got a good kind of expanse across the range here. And we put thermal augers in more than 30 nests and more than 56 nests, but I'm only going to show you the outcome of the nests that don't get eaten by raccoons or wild pigs and stuff like that, which is a significant number. And so what we do is we go find these nests, and we'll stick a thermal auger in the nest cavity. So these little tidbit things, and they take a temperature measurement every 5 minutes. And we put one in the nest cavity. This was one where we were actually collecting eggs, but we try to be not that invasive when we're doing it. And then we stick another logger out here in the micro environment, so close by the nest, not inside the nest. It's usually in the shade if we can do that, and hangs off the ground. And then we have weather station data. These black dots here are the weather stations that we're pulling data from. And from each nest, we get data that looks like this. So this dark line here is the inside, the internal nest cavity temperature, right? And this lighter gray is the microclimate. And then these dots here are the daily maximum or the daily minimum from the weather station that we're getting. And right off the bat, you can kind of see this daily fluctuation here that's occurring, and it somewhat relates to the weather station data. The other thing I wanted to mention too, this will become important, is that once we get this, what we can do is use our understanding of developmental rate, which is affected by temperature. And we can go in and we cut everything out except that thermosensitive period. So this is only looking at the portion of development where temperature is actually informing that sex determination. And so this is all of the nests here by temperature. The temperatures here on the Y and the X are just different nest numbers. And from the 2 different sites, so this bluish purple, I'm really colorblind if I say it wrong, is Kennedy Space Center, and the green is Yawkey. And what we notice, right, is that there's a lot of variation, and that you kind of have to think about the variation across different scales. So you can look across different years, you see variation. There's warm years, there's cool years, like 2014 looks like a pretty warm year, 2013 a little bit cooler, 2012 had some warm mess. You can look within a year. So just within a year, these are the temperatures. So these dots here at 32.5, anything above this dot, we would expect to have a male bias, or this dotted line, and everything below it, we would expect to have a female bias. So you can see, we would see maybe a couple of nests that might have a male bias, and a lot of nests that have a female bias. And then you can look even within a single nest, and you see a ton of different temperatures that get experienced during the thermosensitive period. And what really struck me about this data, coming from a developmental biology background, was that these embryos, so 70% of the nest will have embryos that experience both a male promoting temperature and a female promoting temperature within the span of a single day, right? And that's not at all how we think about TSD, where we just put them in an incubator at this one temperature and go, okay, it's staying there, and that these are going to be males and those are females. But what we have now is this case where you're getting these opposing these opposing environmental cues, and these embryos are integrating this over the course of several weeks to eventually reach some sort of threshold response. So we never see hermaphrodites with a testis and an ovary, that there's some sort of threshold that gets reached. And so we're trying to actually work on that right now to understand, in general, how organisms are taking these transient environmental cues and making decisions with them. So the next step we wanted to do is see if we could actually model some of this and understand what was driving the variation that we were seeing. And so we looked at all sorts of different things here. We looked at the weather station data. We looked at the microclimate temperatures. And what you're seeing here, these are effect sizes or coefficients from this model that we use. We use a modeling approach based off of elastic net regularization, which is useful when you have a lot of different variables and not so many observations, which is the case with basically anything we do with alligators, it seems like. But then we looked at other things like the nest architecture. How tall is the nest? How wide is the nest? What's it made out of? We looked at the architecture of the nest cavity in relation to the nest. How deep is the nest cavity, et cetera. And what I can tell you is we're really bad at understanding what's driving those temperatures within the nest. So we can train a model, and then when we take that model, which is okay, it's like about 40% of the variation is explained. But then when we test it on like a new year, so we do this each year, and we go out and you can test, and you can only explain 10% of the variation from nest to nest to nest. So the biggest drivers was a little bit of the shade was kind of important, local elevation, like how close the nest is to water level. It's kind of informative, but not really. And so that was kind of disappointing to us. But what we did see is that across years, we were much better at being able to understand what was driving the nest temperatures. So if you look across years, what we found is that the mean daily maximum temperature was the best predictor of nest cavity temperatures by a long shot. And so this is all of the years that we've measured, and this is the mean nest temperature, and this is the mean weather station temperature. And what we saw are these, just turns out just a straight linear model is our best bet. And we can, at Kennedy Space Center, explain 80% of the variation, and at Yawkey 72% of the variation. And there's this nice positive correlation between how warm the environment is in these nests, although there is variation within each of these years. And so then what we did is, okay, well, let's just take this linear model and see what happens when we take in all of these different climate scenarios. And so we use these models that everybody else is using. There's several models, so you have some level of uncertainty there, 30 models. And we use these two emission scenarios, RCP 4.5 and 8.5, which is just based off of how policy will be enacted, or really how many greenhouse gases are emitted. So 8.5 is kind of worst case, 4.5 is middle of the road. And I'm not an expert on understanding like where we are in relation to this right now, but maybe some of you are. But then what Samantha was able to find is this statistically downscale model. So usually the resolution of these 30 models that go out and predict into the end of the century have these really large, kind of coarse resolutions. So I think it's like 25, 30 kilometers. And you can statistically downscale that to about 1 to 5 or 1 to 3 kilometers. So we were able to really get more specific predictions for our field sites here. And this is what nest temperatures we think would look like based off of the data that we have, where you'd see mean nest temperatures here. And you generally see this increase starting now. And then around 2050, you would see where the two emission scenarios start to diverge there. And so what we then did is take those estimates and run them through our reaction norm that we have for sex determination in the alligator here, and translate these into sex ratios. So this was is at Yawkey here, and this is the proportion of males, and this is the year. And what you can see is that we're expecting, like, should be happening now, is a really marked increase in males being produced. And then you see, around 2050 again, you start seeing almost 100% males at NASA or Kennedy Space Center. And then depending on that emission scenario, you'll either, in the best case, you see, lowest emission scenario, you see the kind of maintenance of these male skews, but at these warmer temperatures or warmer emission scenarios, we would see the production of these high temperature females, right? So it's basically following this reaction norm here. What you're not seeing here, and this is one of the problems with the conversion to the PC, is all of the uncertainty here. So the error bars for some reason are missing. But this whole area is like uncertain, and to be transparent about that. And the reason is, we think, is because each one of these models differs a little bit in the temperature, right? But small changes in temperature due to this steep reaction norm have really dramatic effects on sex ratio. And so it's kind of uncertain, but these are the trends we might expect. So I'm going to come back to this towards the end of the conclusion. What I can tell you is that nobody right now is looking, and all crocodilians have this same general pattern. No one is actually monitoring sex ratios in hatchlings right now. So we really have no way of verifying that. And one of the reasons is hatchlings aren't sexually dimorphic, and it's really hard to determine what sex they are. I'll come back to that towards the end. All right, so this comes back to this one schematic that I showed, right, where you've got this conservation and divergence. And it really begs this question of why use temperature to determine sex in the first place? Like, why is that a good idea? What would be the evolutionary advantage? And we have typically spent a lot of time on the proximal mechanisms, really trying to understand how this kind of physical abiotic factor gets translated into these biological genetic cascades. But through a series of events, we've also become interested and maybe got some traction to try to understand some of the evolutionary drivers of temperature-dependent sex determination. So, and I'm going to share some of this work is kind of ongoing, but I think it's cool, and I think we're finally getting a little bit of traction here. So most TSD species are really difficult to study, right? They live a long time. So turtles live a long time. Alligators live 60 years or more. And so it makes, measuring fitness, for example, really difficult to do. And despite that, we have a lot of theory about why TSD might have evolved. And if it's just neutral or if there's any sort of adaptive value to it. And the real kind of pioneering work, some of the first work that really came out with a solid and clear theory was by Charnov and Bull. And there's some caveats here, but the main kind of premise that they have is that you would have TSD to be favored when you have temperature that influences the sexes differently, right? And you're seeing that here. So this is the fitness on the Y. This would be incubation temperature here, cold and hot. And in this case, what you can see is that the fitness of females has a steeper slope than it does for males. And so a female produced at a warm temperature would have a greatest fitness differential compared to the cooler temperature than the males. So that's what the kind of the real meat of that theory was. And since that time, you've had all of these other models that have been proposed. They all contain this differential fitness component, but they kind of vary in the mechanism that gives you the differential fitness. So one of them is matching sex to the timing of hatch. And so in this case, it's incubation affects the timing of hatch, which affects males and females differently. And there's actually some empirical support for this. This was work by David Conover in 1984 on the Atlantic silver side, so all up and down the eastern seaboard. And they have TSD. And females are produced under cooler temperatures that are more reflective of early spring temperatures, right? So it's like early in the year, cooler temperatures, you get females. Later in the year, it's warmer and you get males. And what they showed is that these females that are hatching earlier, these fry, they have a longer growing season, and female fecundity is more tied to the body size than it is in males. And so these females have an advantage, and if they have this long growing season that's not quite as strong for the males. So you get this direction there. There's this other model here, which is a sex by temperature interaction. And briefly, this is just where you have incubation temperature that actually affects male and female development differently. And that leads to these sex bias fitness consequences. So one way, I guess, to think about that would be that if you had incubation temperature, like a testis or an ovary that develops at a female promoting temperature would be a better ovary and lead to greater fitness than an ovary that develops at a male promoting temperature, would be the way to think about that. No empirical support for that. And then the other is kind of the counterpart to the sex by temperature interaction, and it's matching sex to phenotype. And so in this case, what you have is incubation temperature affects a phenotype. Doesn't matter what sex, just affecting a phenotype, right? Regardless of sex, but that phenotype that's affected ends up having sex bias fitness consequences. So that's kind of a lot to get through there, but it's kind of necessary for where I'm headed. So I'm going to focus on this matching sex to phenotype, right? Where incubation temperature affects phenotype similarly for males and females, but phenotypes convey sex-dependent fitness consequences. And so Lisa Schwanz, who's pictured here, she's got a lab in Australia. In 2016, made a really acute observation. And what she observed was that many TSD species have a tremendous difference in age at maturity across the sexes, so that you see male and female individuals reaching maturity at very different ages. And she was kind of like, why is this so pronounced? And particularly, why does it seem so pronounced in TSD species? And based off of that observation, she kind of extended this matching sex hypothesis to this survival to maturity hypothesis, which is that incubation temperature affects juvenile survival. And when age at maturity differs across the sexes, the later maturing sexes produce at optimal incubation temperatures. So to distill that down, there's really just there's two criteria that need to be met. Sexes display differences in age at first reproduction, and that incubation temperature affects juvenile survival regardless of sex. So she likes turtles, and this is a schematic from her paper here. And so she's got these are little baby turtles here. But this is the general idea. So you've got these eggs, and if they're incubated in a hot patch or warm temperatures, you'd see 95% annual survivorship. And at a cold patch, you would have 85% annual survivorship. And so now temperature is influencing survival regardless of sex. But here, the males are maturing at year 3, and the females are maturing at year 6. So the males don't have to survive as long to reproduce, whereas the females do. They have to survive twice as long. And so the idea is that under this case, what you would want to do is have the sex that is maturing later produced at these temperatures that gives you optimal survivorship, because they have to survive longer to reproduce, right? So that was the general idea. And right at the time when Lisa published this paper, we had a master's student in the lab who was doing some parentage genetics. So alligators generally we think of as reaching physiological maturity at about 7 years of age, maybe 8. So physiological maturity, people have looked at, like, the way we know this is people would actually go out, capture a male. I'm talking about males, go out and they would use an index card, like you would make flashcards with, fold it over, and they would the alligator penis is this long structure, and it's got this slit that runs all the way down the middle, and they would run this card down the middle of that, what they call the sulcus, and then put it on a microscope slide, and they would see sperm. And so they go, that's a mature alligator, right? So I don't know how that would have passed the eye cut right now, but anyway, it seems painful to me. But anyway, glad I wasn't one of those subjects. So the other, so females, they reach age at first reproduction at around 14 years, and this is work from Phil Wilkinson and Thomas Rainwater as well, where they trap females at the nest. So this is pretty good, right? It's like trap the female at the nest, and then we had this master student, Josh Zadel, who would take a few eggs and could confirm that these females that were being trapped at the nest were actually the mom. And what we found that was surprising, though, is this is at Yawkey, and you've got this really great historical data set where we know how old these individuals are. They've been captured since 1979 in Mark. And what Josh found was that males don't actually start siring nests. This is all the males that we had in our library, and this is how many nests they actually sired. And what we found is that they don't actually start siring nests till they're about 24 years of age. So they could make sperm age 7, age 8, but they're not actually reproducing until almost a decade later than females. And so based off of this differential age at maturity and the alligator reaction one, we go, okay, well, maybe according to Lisa's hypothesis, we would predict that these temperatures that produce males should give us the best survivorship, and compared to these temperatures, both low and high, that are producing these females. So that was the general idea. And we went and we looked and we go, okay, well, what do we know about juvenile survivorship in alligators? And we don't know much at all. What Samantha did, and was she started combing through the literature to find all the papers that had reported sex ratio data. And particularly, they had to have sex ratio data, and they also had to have size class data. So like what size classes they were looking at. And I'm summarizing that data there. Let's see. So these are hatchlings here. This is juvenile and this is adult. And so if you looked at hatchling sex ratio papers, they were all female biased, all of them. And yet all the juveniles were male biased, and same with adults. And so this had been reported previously that, you know, why are adult sex ratios male biased? And I can tell you males and females are different sizes. And we had a lot of skepticism about this data, because if you put you put somebody out there that with a snare trying to catch an alligator, and you say don't pay attention to size, we just do it as unbiased, they always go for the biggest alligator. And so like there's no way we really especially believe all of this, but we did. This is what it was. But anyway, there looks to be this big jump here, and it's not just in South Carolina, right? This is the same data that just showing that where all these studies came from. And so what you see is the shift that takes place between hatchlings and juvenile. So the next thing we did, and let's see. Got it. Let's do this. All right, so what we wanted to do is actually go out and see if we could assess survivorship ourselves, which has been a real learning experience for me, but it's been a lot of fun as well. So the first thing we did is we went to right out our back door to Parr Pond. This is at the Savannah River Ecology Lab. And we just collected 4 clutches of eggs. So small kind of pilot level. We incubate those at the male promoting temperature of 33 and the female promoting temperature of 30 degrees. So not any high temperatures here, just 33 and 30. And we release all the hatchlings that hatch, and we go back every 3 or 4 weeks and we do these kind of exhaustive recapture efforts. And this was the part I was really skeptical about, is like, if we release these animals, would we ever find them again? And I was very doubtful, but it turns out we could. And they generally stay in the same area, and that's been reported before. That wasn't new. But if you actually look at the survivorship here, and we've done this several different ways. I'm old school kind of experimental guy, so I like just seeing the recapture numbers here. But I know everybody here, and Samantha too, is like, no, you can't do that. You have to mess with the probability. And so these are the Cormac Jolly Sieber survival probabilities. But what we see, regardless, is that you see male promoting temperatures convey this increased survivorship relative to the female promoting temperatures. Those are low numbers, right? I think this represented something like 10 animals or something like that. 1 bar, and that might have been 20 or something. So not a ton of bars there. And we can break this up into pre-winter and post-winter. So then I said, wow, well, that's pretty cool. And we don't know much about juvenile survivorship. So let's keep going with this. And Samantha was chomping at the bit to do a bigger experiment. So we did this one at Yawkey with Thomas. And the nice thing about Yawkey is there's a lot more alligator nests. So we scaled this up to 8 clutches, and we incubated male and female promoting temperatures. But this time we used hormone treatments to sex reverse. So the problem is in TSD species is when you incubate at a male promoting temperature and a female promoting temperature, you've got different temperatures, but you also get different sexes. So it's really difficult to deconfound incubation temperature from sex. But what we could do is incubate at male promoting temperatures here. And we can actually just pipette a single dose of estrogen at a particular developmental stage onto the egg. And that sex reverses at 100%. So you get this 100% sex reversal to females. So that way we had individuals that had been incubated at 33 degrees but yet we're phenotypically females. So we release all of these guys, and we continue the recapture work. And we see the same thing. So in this case, what you're looking at are the two male promoting temperatures here. The green is the sex reverse, so these are phenotypic females, these are phenotypic males, and these are females either with the estrogen or without, but in any case, they're both females, and estrogen itself doesn't seem to have any effect on survivorship. But we still see this both pre-winter and post-winter, and those are the probabilities there. So I know I'm kind of running out of time here, and so I'm going to kind of hustle through. I think this is close to the last slide. The last experiment we did, so we've been doing this kind of every year, is we've shown that we get consistent differences in survivorship across these two temperatures, right? Low and intermediate, but we still didn't know about this high temperature females. So that year we did the same experiment, and I'm just showing that data in 2021 here. And what you can see, this is the high temperature. This is these should have a female bias. These are the male promoting temperatures. And again, we see this difference there that actually starts to resemble the reaction norm there. And you can see the probabilities are the same way, which together is kind of supportive. So the question now that we're working on is trying to understand, well, what is the organismal biology that is linking incubation temperature to differences in survivorship, particularly within this first year? And what we had noticed is that if you look at offspring size, there's obviously this nice correlation. This is hatch mass, this is egg mass, between egg mass and hatch mass. But this is from an experiment where we incubated at all sorts of different temperatures here. What we noticed is that at the warmer temperatures, they seem to be a little bit larger than expected, and the cooler female promoting temperatures a little bit smaller than expected. And that's what you're seeing here. It's just these are just the residuals off of this line here. Where at FPT, you can see they're generally smaller than you would expect, and at MPT they're larger than you would expect. So it's like, okay, so maybe it's this developmental efficiency thing where these maternal resources are most efficiently converted into hatchling at these intermediate temperatures. But we didn't know. So before we released all those hatchlings, we had measured and taken pictures, and so we had all of these traits. And when you look at this is SVL on this row across all 3 years. Mass, this is snout vent length, so just a measure of length. This is mass, and this is body mass index. And these are not like whopping differences here, but you generally see not real strong effects of length, but on mass, the males generally appear heavier, similar to our previous findings, and the body mass index is generally larger, and even with those high temperatures. So then we look at survivorship to see if these traits correlate to survivorship at all, or survival probability. And this column here is all the years put together, and you see these kind of weak but positive slopes. It's like, okay, maybe. But then if you look across each years, what is wild is how much variability you can see. Like in 2019, SVL looks like it's super correlated to survival probability, but in 2020, it's got like this negative effect. And 2019, again, you see these steeper slopes. And so we're not quite sure what to make of all of this. If you look at the literature, there's these ideas, right? The bigger is better. And people have tested this in all sorts of different species. And what you do see, though, that is quite revealing to me at least, is that I've seen lizard papers, there's some bird papers, and a lot of the times it's context dependent, right? So it's not, I don't think that the size isn't important, but I think it's probably very much dependent on the underlying habitat that they're released into. Maybe it's the weather environment, the harshness of that winter. I think it's probably the reason we see variation there is I think not that it's just a bunch of noise. I think it's probably very much dependent on, you know, ecological and environmental dynamics that are occurring there. But in any case, we're still trying to nail this down. We videoed some of these guys to do behavior studies to see if that correlates. So we're still kind of this is as far as we've got right now. So I'll just kind of conclude here really quick. So the efforts, I think in terms of conservation, we really need to understand hatchling sex ratios, and we don't have real good data for that. There's only a handful of studies in alligators, but also other crocodilians, including a lot of other turtles. And one of the big problems with doing this is that hatchlings, the external genitalia are not that different. You can't really feel good about the call. You look at it, and just you don't feel confident on even looking under a microscope. I don't. And there's no sex chromosome, so you don't have any genetic markers. And so we recently tried to use the DNA methylome of red blood cells to see if we could use methylation marks, because we know that gonads have very distinct DNA methylomes. There's just a chemical mark on the DNA. And what we can do is build these models, and we can actually determine whether the hatchling is male or female pretty good. And what was even more kind of interesting to me is that to some extent, we're able to actually understand if that individual, what their nest temperature looks like. So the idea I think we need to do is go back now to certain populations, or maybe alligators, not, you know, some vulnerable populations of vulnerable species, and really start looking to see, you know, are we going to experience these male shifts? I showed that incubation temperature, but not hatchling sex, influences survivorship. That temperature-dependent phenotypes that mediate the effect of incubation temperature on survival are complex and likely dependent on ecological and environmental dynamics that we're not measuring. So I would really, I think what we needed, we don't know how these hatchlings are dying, the ones that we never recapture. Whether it's starvation or predation, don't have a real good idea about that. Have a sense that there's definitely some level of predation. Don't really know how many are starving, but when we see these guys at spring, they're like the first spring after, you know, winter, they're just skeletal. I mean, they're alive, but they're just frail. And then shortly after that, they bulk up. They have these, you know, distended bellies. They're just really full. So I think that's a big bottleneck that we need to pay attention to as well, maybe from a metabolic perspective. And then together with previous observations, with this differences in age at first reproduction, our data support the survival to maturity hypothesis regarding an adaptive value of TSD. Still need to do work on that. Then I'll just thank a few people. So this is Samantha again. A lot of the work I showed is hers. We couldn't do this work without the support of Yaki and Thomas. And all the other people in the lab, and our funding has been instrumental as well. I'm trying to think. And then Russ Lowers at the Kennedy Space Center, who does a lot of the field work and trying to get these thermal loggers deployed out into as many nests as he possibly can. And Yaki and SCDNR for permitting and support as well. With that, I think I'm done.
I think we've got time for a couple questions. Yes.
So I want to ask about your predictions with I think the next 50 years, I think mostly like male populations of alligators. So what about the reverse? Like during, say, like the Ice Age, I mean, alligators been around for like millions of years. So somehow they made it through that. And even like the southern parts of the equator, I guess that's where they may have relived it in theory. Would there be mostly female alligators? Probably, and then I guess building off of that, do you think with incubation that nests and their materials change, like they vary at all to maybe have cooler or warmer temperatures?
So I'll start with the last part first. So the last part is about whether there's differences they can compensate at all, right? Either through changing the architecture of their nest. I see that shade, they build their nest in a shady area that has a bigger influence than what the nest is actually made out of. But what we did look at is to see if in warmer years, would you see a higher proportion of shaded nests? And we don't see that. Indicating that their maternal behavior, at least in shading and elevation of the people we looked at, are not sufficient to compensate. I think hatch date might be the next technology, whether they nest earlier or later, might be one other way that they can leave. Again, don't see in warmer years that they're nesting earlier than cooler years. So it doesn't look like there's some sort of signal that they're necessarily responding to that's a thermal cue. And maybe it's photo period, I don't know, but it's not like really warm years, you get a huge shift in early nesting versus late nesting. Which brings up your question, which is how in the world do these organisms persist over really long evolutionary timeline? I don't have a great answer to that. I mean, I hear that a lot, that question. I think a lot when you talk about ice ages, I think there's a lot of things happening, right? So that extend beyond just yes, but I think range expansion. So I know like the northernmost crocodilian fossil bones and stuff that have been recovered were in Greenland. So which is really far north, right? And so my guess would be that their range shifts and changes over those timelines. I don't see how that's going to work with kind of all the impediments that we have. I just think about like some of the, if you Baltimore or somewhere on the Atlantic seaboard, I don't see them like welcoming a horde of alligators, and maybe, so I think I don't have those answers. I think depending, you talk to, it seems like there's an inverse relationship with who you talk to in their age. Like some of the kind of older crocodilian biologists, they're like, oh, they've been around forever, they'll figure it out. Maybe they will. I have no idea, but nobody's even looking to see. So I think there's some opportunities to study them. And I don't know what will happen.
Good question for you, Ben. So I think that last part of your talk is super interesting, trying to figure out what that linkage is between sex and survival. Do you have any plans to look at, or is anybody looking at endocrinology, like stress responsiveness or behavioral phenotypes of individuals hatched with those different cultures?
Yeah, so we had a student in the lab who was doing these behavioral assays where he basically puts the hatchling in a cattle tank about the size of this table and puts basking, makes one side warm, tries to measure all these behavioral traits. And he was looking at mercury that is maternally deposited to see if that influences. But he also incubated at male and female promoting temperatures. And that was by far the biggest influence on hatchling behavior relative to mercury. It was a big effect. And most of that was with, see if I can remember. He had some measure of boldness, which was like how they kind of track along the side and their basking, how willing they were to bask. And so those were different across the sexes. Anecdotally, and I'm not sure how much experience you have with this, but like the females are FPT. You grab those and they want to bite the hatchlings, and they'll like do a little death roll. Whereas the males, you can touch them on their snout a lot, and they don't ever really open their mouth. And so I don't know what that's about, or if for me, it's like, what would be the behavior that would determine survivorship or influence survivorship? I'm not sure I have a great handle on that, but I think we did. You know,
Maybe like freeze response, like almost kind of.
yeah, he did that. Yeah, so he had this this great blue heron. It was like a yard decoration, and he had a pulley, and so halfway through, and it would change, but halfway through, so either before or after this, he would pull up this heron, and the hatchlings would freeze for a bit, and then he measured that, and I think that was temperature dependent. Yeah.
With that, like your age at maturity for alligators, do you think that there's a behavioral component to that? Because males earlier on in their lifespan will not be territorially dominant in a lot of habitats, particularly in areas with higher densities and the rest of their range. So I wonder if some of that, you know, like age at sexual maturity for the normal populations may have to do with like body size and also just like behavioral components, like how are they able to compete within those systems?
I think that's a great question. And I would be really surprised if we did not see variation in that age of maturity or age of first reproduction across populations. So Yawkey is interesting, right? It's these old impoundments, it's on the coast. So you've got limited fresh water, but tons of food. Got a pretty nice population there, but alligators don't have salt glands, so they have to osmoregulate and get in this fresh water. So one of our you also receive really low genetic diversity at Yawkey. And so one of our kind of ideas is that at Yawkey, you see these large number of alligators that become really densely packed in there during certain periods of the year. And perhaps a male's shutting down reproduction for a lot of the other males. And so from an endocrine perspective, there's been other work that have looked at alligator testosterone levels throughout the year, and they looked across size classes. And so about this time of the year, you see both in smaller males, subadult males and big males, you see their testosterone go up. But right about April, the smaller individuals, their testosterone levels just crash down, right? And I think that's probably mediated by some sort of aggressive interaction that might be influenced by density. How many of those individuals kind of get shut down? Presumably there's some trade-off with high levels of testosterone that it's not worth to keep it that high if you're a small male. Yeah, absolutely. And then how that might influence the evolutionary dynamics of this sex to maturity hypothesis is real. When you have a smaller difference, how does that translate into the selective pressure that's actually driving the sentence? They're really good questions.
So I want to talk about careers. So back in careers, I think they have very low skilled male ratio, like 10 to 90 maybe. So this putting those in incubators and promoting the temperature to determine sex can be a like milestone in conservation. So can you tell me a bit about what about the lab setup? How feasible is it to do practically over like our budget for us?
Well, I think incubating eggs is really easy, and your limiting factor is like the incubator. So it's pretty straightforward to do. The aerials are even easier, right? Because they kind of have these communal nesting grounds and the bends of the river and those beaches where a lot of females lay in close proximity. I don't know how easy they are to find, but I think mitigation efforts might not be that difficult. And we've released hatchlings. When I was at postdoc, one of the first experiments, I ended up releasing a bunch of hatchlings at Yawkey in 2012. And when we go out to recapture, Thomas and Samantha and myself have seen those that we've recaptured. So they will survive. It's not a bunch of them, but I think it's completely doable, but we just have no idea if it's an issue at all, right? I mean, this prediction we have is a pretty crude model to be predicting these, but it's the best we have. And so we just have no real ground truthing at this point. So I think that's the first step, is to see if we can ground truth and see if we see skews, or is there some sort of maternal compensation with nesting that we're not thinking of.
You said like you released 115 nestlings at the first, so how many did you recapture those to monitor the mortality?
So for the males, 20% will make it through winter, their first winter at that MPT, right? And like less than half of FPT will do that. So is that what you're asking? So you'll see about 20 males if you release 100 that will make it through their first winter. After that, we think dispersal becomes an issue because we're releasing at some of these same places, and we're not. We'll see some, but not a lot. So it seems, and that's on record. Someone's dissertation, can't remember who's, but looked at dispersal. And they generally stay around the same place for about a year, and then they start to disperse quite broadly.
All right, thank you, Ben.
Thank you.
Good. And we have lunch coming in now. If you signed up, so you can stay and continue to chat and ask questions. Remember, the slide, Laura. She was supposed to keep our house.
Oh, yeah.
Lots of family, you're gonna do a PhD case. So it's just about all that main channels. She's like pushing. So she's. Yes. Sorry.